chloroplast, there is a thicolloid. In the thicolloid membrane, there are a series of proteins known as photosystem 2 and photosystem 1. In photosystem 2, there are electrons. Light energy enters photosystem 2 and charges the electrons. These electrons move down the electric transport chain. The electrons that were in photosystem 2 are replaced by electrons from water. As a byproduct, oxygen gas is made. The electrons removed from water are used in a protein pump in the electric transport system to pump hydrogen into the thicolloid. This creates a concentration gradient which powers the protein ATP synthase, a protein which turns ADP into ATP, like this. The electrons used to take in the hydrogen are moved to photosystem 1. Light energy enters the system and charges the electrons. The electrons are then transported to another protein, the electric transport system, which is used to reduce NADP plus to NADPH. NADPH enters the stroma, along with the ATP from the ATP synthase. The stroma is the fluid inside of the chloroplast. These reactions are called the light reactions. In the stroma, ATP and NADPH from the light reactions are used to fuel the Calvin cycle. In the Calvin cycle, CO2 is joined with ribulose bisphosphate, also known as RUBP, to create a 6-carbon molecule. In a process called carbon reduction, the 6-carbon molecule is turned into 2 3-carbon molecules. In step 2, the 3-carbon molecules go through carbon fixation. Carbon fixation is fueled by NADPH and ATP from the light reactions. NADPH goes in, NADP plus goes out. ATP goes in, and it gets turned into ADP. After this process, the 3-carbon molecules are turned into glyceraldehyde 3-phosphate. In step 3, RUBP is regenerated. After 6 cycles of the Calvin cycle, 6 molecules of G3P have been created. 5 go through the cycle, or continue going through the cycle, to be regenerated into RUBP. ATP assists in this process, going into the molecules as ATP and exiting them as ADP. After this, 3 molecules of RUBP are made to continue the cycle anew. The remaining G3P is joined by another G3P. Those 2 G3P join together to make glucose. The phosphate of the glucose leaves the glucose and goes on to make starch and cellulose. In the absence of that phosphate, fructose can join in and combine with glucose to make sucrose.